Okay, Matt gave a great overview of all of NIH, and so I'll try to be not repetitive in going through this today. I'm going to talk about translational space, and the space I'm really going to be in is more that bench to bedside translational, where our programs are located. The Division of Clinical Research focuses on the later stage clinical trials, and I'll just touch on what those opportunities are with one slide at the end. A disclaimer, all the opinions you hear are my own and not necessarily those of the government, so, and I have no financial conflicts of interest. The agenda I'm going to go through today is just a brief introduction to NIH Neuroscience, and then go through the Division of Translational Research, some of the big programs, the IGNITE program, which is our feeder program, the Create Biologics program, the Blueprint Neurotherapeutics Network, and then touch on, as I said, translational neuro devices and clinical resources. And then I'm going to go through what I know about other translational services that are available in other ICE institutes and centers that might be of interest to people in the neuroscience space. And lastly, given if we have some time, I'll talk about some general considerations when you're applying. So introduction. As Matt pointed out, we're the largest biomedical research institute in the world. This is the clinical center at NIH campus. There are 27 institutes, all of which have different missions, different funding priorities, different budgets. And so it's really important to know your indication area. I stress figuring out who your program director is at your IC, because they can be your best friend from the disease side as well as my partner on the translational side throughout these projects. Neuroscience funding, much like you saw before, is also spread across many institutes. And the big three institutes for neuroscience funding are NINDS, the Neurological Disorders and Stroke, Mental Health Institute, and Aging. But also big players are things like NEI and NIDA and NIAAA. So we cover a large swath of neuroscience across many different institutes. And in fact, neuroscience funding is the largest sector of funding in NIH. Another important thing to recognize about grants at NIH is you'll see R grants, things like R01. These are the most common grant application program. They support discrete, specified, circumscribed research projects. And you're sort of on your own in those cases to go off and do the work. And then there's the U grant mechanisms, cooperative agreements. They are an assistance mechanism in which you're going to have substantial NIH scientific and or programmatic involvement in the project. And the project for which I'm responsible for, the Blueprint Neurotherapeutics Network, that means you meet with us every 2 weeks. So it can vary, and you have to really look at and see what's going on. And I'll explain why that's true when I get to my program. So the U44 is the SBIR version of this cooperative agreement, and you'll see that throughout our presentation today. You'll also notice that, as Matt pointed out, some funding opportunity announcements will have PAs, PARs, RFAs. They all mean different things. They can have different requirements for review, different requirements for receipt dates, different requirements for all sorts of things. Make sure you read them carefully. This is the number one thing to take home, if nothing else. Read the FOA carefully, because failure to do so can end up with your application getting bumped out of the queue before it ever gets reviewed. So ask yourself, is it a cooperative agreement? Is it milestone-based? Is it an SBIR mechanism? And make sure you follow those instructions. There's a wealth of information online, and I give a link here for a starting point if you've never applied to the NIH before as a good first stop. The SBIR has another website that's also a great first stop if you're coming for SBIRs. Who can answer your questions at NIH? Before you submit the application, the program officials, program officers at Institute, or a scientific review officer, pretty much anybody you can get information from before you submit at NIH can help you out. After you submit, before it's being reviewed, you are now in the realm of the scientific review officer. So if you have a question like, I want to add something to my application, The SRO is the one that's going to determine whether that can be done or not. After the review is done and you've gotten your summary statement back, you're now going to come to the assigned program officer. There's going to be a single program director that's listed in your area on the FOA. That's the person that's going to be responsible for determining whether that project gets funded, in part by defending it to institute management and supporting it through council approval. So that's at least the starting point for knowing where you need to go and who you can talk to. I'm going to get into sort of our division now. And the Division of Translational Research has a mission, and it's really to accelerate basic research finding towards patient use in neurological disorders and stroke by providing funding, providing expertise, providing the resources necessary to the research community. And our website is here. All these slides will be available to you. So we have a portfolio of projects that we fund through the Division of Translational Research. I'm going to talk about the ones that are in sort of that yellowish color, but I want to at least touch base on the ones in blue. The epilepsy screening program, therapy screening program, is a free of charge service where you can get in vivo testing of your molecules for seizure models. You have to submit it to the program. They're tested through a contract at the University of Utah. And this program has been going on since the 70s. And many of the therapeutic agents that are on the market for epilepsy today had some ties to this program at some point, or its predecessor, the anticonvulsant screening program. The SBIR program, STTR program, I don't need to touch. You just had an hour, so I won't go through that. The Countermeasures Against Chemical Threats, Counteract, is a program that is a joint program. It's a trans-agency program. That's this particular group is on small molecule type work, and it's to have countermeasures against the release of chemicals at, for instance, at a chemical manufacturing plant. It could also be for antidotes for other things. So with that, I'd like to sort of go into the other programs I talked about in the beginning. So the first program out is the IGNITE program. The IGNITE program is a program that we view as a feeder program in the Division of Translational Research. If you're coming into an NINDS program down the road for biologics or small molecules, you may need some things. You might need assay development and therapeutic agent identification and characterization. So things like, I need to get an assay up and running. I need to be able to run a high throughput screen. I might be able to resynthesize that molecule and see that it's really a validated hit. That can be accommodated through this PAR. I really need that preliminary data you talked about before. You need that piece of preliminary data, the pharmacodynamic read in vivo, efficacy studies like that. I just need that one piece of data. You can get it through PAR 15071. These are milestone-based programs. They are an R21, R33 mechanism, and I'll go through that a little bit more in a minute. The last one is I want to develop and validate a model for drug discovery, an animal model like that. In all these cases, Marianne Pellemonter is the program director at NINDS and should be the person you would contact. The purpose, as I said, this is an R21, R33 mechanism, and the first part of it is you have to demonstrate feasibility and prepare for the R33. It can be $750,000 across the whole project, $250,000 a year to do this, but you must write in a clear go, no-go milestone, and does it warrant the further investigation to go to the R33 stage. If you have questions about this, Marianne can certainly help you. The program officers can help you with this. Our biologics program is called the Create Bio program. We fund things like peptides and proteins, oligonucleotides, gene and cell therapies, and all fit into this program. And it's broken into two tracks. We have the optimization track and we have a development track for this program. So in the optimization track, it's really all about getting the molecules ready for the IND-enabling studies, doing the preliminary work required to get that done. characterizing your lead candidate fully so that you'll do well in review when you get to the development phase. The development is all about submitting IND applications and conducting phase 1 clinical trials. They have some other resources available within this program as well. They have some consulting so that you can work through them on CMC development consulting, on regulatory affairs consulting, and statistical consulting if you're in the program. So they have some resources they can bring to the project from within NIH that's not part of your actual grant budget. They also have a number of related resources like the application support library and the stem cell information. And they also interact regularly with the FDA and can provide some guidance to you on what they learned through that process. There are new FOAs, just listed as Matt pointed out. Almost all the FOAs have been reissued to deal with clinical trials in the last few months, and so you'll see many listed with PAR 18 now, and so the Crate Biodevelopment have just issued within the last couple of weeks. As of February 20th receipt date for that. Note that the optimization track, those have not been issued yet because there's no clinical trials there, and the February 13th is the receipt date, so it's a little different date. Read your FOAs carefully because the dates do change. The Blueprint Neurotherapeutics Network, which is my program, and I'll talk more about this program than any others, is unique in the NIH. It is directed at small molecules. It's a trans-NIH project. It provides funding and necessary resources in terms of control, access to CROs and drug discovery expertise. What's really unique about it is our CROs have what's called determinations of exceptional circumstances on the MedChem and the drug manufacturing formulation contract. What does that mean to you? That means the intellectual property that they create, they assign back to you. So that in the small molecule space, the intellectual property is staying with your institution so that you'll be able to license this program down the road, even though you're using our contractors. The purpose, de-risk the project. We want to get it to the point where industry invests in you and advances this project to hopefully help patients efficiently. As I said, it's a trans-NIH program. So that means we have 9 institutes that have signed on. All 9 institutes have signed on to both the non-SBIR and the SBIR track. So you can go to any of these institutes if you're within their mission and talk to their program directors listed here and say, hey, would you support a program in small molecule space through the Blueprint Neurotherapeutics Network? Again, look carefully at the FOAs. We're going to take your ideas, your small molecule starting point. You must come in with a small molecule starting point. Your strong disease assays and models. You've got to have good preliminary data and hopefully have some novel drug target, although you don't necessarily have to know the drug target. It can be a phenotypic screen. I should point that out. And we're going to couple you with industry expertise. You have advisors with extensive pharma experience that we add to your project team as you go through the project. And we have industry standard contract services. None of these are included in your budget. They're just part of what you're going to get if you're a successful applicant to the program. And our goal is to get to first-in-human trials. So you could also enter in anywhere along the spectrum. So it doesn't have to be, I just got a hit out of a screen. It could be, I've already done some optimization. I'm really got maybe a year or two of chemistry left. I want to come into the discovery track at the hit to lead stage. As long as you can say, I can define a go, no go milestone for that first phase, that UG3 phase, which is 1 year, or the U44 phase, which is up to 2 years, that you can truly define it as a go, no go point, we're probably good, and we can come in and do that. Some can come in as late as the development stage. I've already got everything, all I need to do, I'm not going to do any more medchem. That's how I define development stage. As I'm done with medchem, I have molecule or molecules that I know. And I'm going to get ready for doing IND enabling. The program will only support one compound through IND enabling studies, and it doesn't do backups. We're really about critical path moving you forward. But not all ICs will accept it. For instance, NIDA, will not accept a development project. NIMH will not accept a development project. NIDA has a number of FOAs and contracts already set up in their organization to support development, and they prefer to use those contracts and their FOAs for that particular aspect. As I said, because it's both tracks, both SBIR and non-SBIR, the UG3, UH3 will take small businesses, people, if you don't qualify for SBIR and you're a small business, you can still come to UH3. You're a foreign institution, you can come to the UH3. You're going to have to justify why you have a unique project, but it can be done. The U44 is an SBIR. You must qualify for SBIR rules. All that applies. One of the advantages that you may find with this program versus some of the SBIR at some institutions is our CRO contracts are not part of your budget, which makes you meeting the requirements for bringing the money in and staying in your organization somewhat a little easier to navigate. The same rules apply. Their guidance in there, and we say up to 500K in phase 1 and up to $1.5 million per year in phase 2. Total costs. If you don't have to ask permission for that, you can put it in. But if you're coming into the UG3, UH3 side, and you're more than 500K in direct costs, you have to ask permission first. And you have to ask permission like 6 to 8 weeks in advance. So you're not going to be able to do that for the February receipt date. If you're going to have that kind of budget, you're going to have to probably go for the August date. What do we have for an infrastructure? Well, between the grants and the contracts, if you're successful in all your milestones, you're probably going to get up to around $10 million in funding from us that we're going to put out between the contracts and the grants if you were to go all the way through phase 1. What you have to bring to the program is the disease biology, the ability to run the disease assays. That means the primary, secondary cellular assays associated with your disease biology you have to have. By you have to have, meaning they have to be in your budget. You can be a virtual pharma and come to the UG3, UH3 and say, I have this CRO that's going to be running this assay for me, and here's my plan, here's my starting point, and I have had grantees and applicants that have that. But you have to bring the disease biology. You don't have to use our CROs. We recommend you do, but you don't have to. If you have an existing contract in place, you can continue with that contract and we'll work with you. But what we can support is medicinal chemistry. This contract is presently up for renewal, and it provides not only medicinal chemistry support in terms of actual synthetic chemists, it also provides the in vitro ADME, running solubility, running microsomes. All of that work can be done through there. And they can also do that work alone to support your medicinal chemistry. So you come in and say, I already got medicinal chemistry going in my thing, but I don't have that in vitro ADME work. Can you do that and support our program? Yes, we can do that. We just can't do one-off experiments. Don't ask me to put a whole contract in place for a single experiment. Just too much money, do that yourself. But if you want to run a program through our contracts, we can certainly do that. PK, running both rodents and large animals, we can do. Running GLP tox studies, we do. Drug manufacturing formulation, salt screen polymorph work, formulating it for API in capsules or things. We do all of that through our manufacturing process. Every project team that is doing medicinal chemistry will put them through have access to our relational database system so that you can put your data in, share it with the team. Each project is put in their own separate vault, so they're not sharing information across teams. And eventually, hopefully, we'll get to clinical trials through our clinical trial network. As I said, intellectual property is retained by the PI's institution. We put together, ex-pharma executives who have experience in medchem and regulatory affairs and tox, all those types of disciplines. We can add to the team the LDTs. We meet every 2 weeks as a team on the phone, do video conferences, and we direct the project going forward. It is you as a principal investigator and the lead consultant that really drive that program. NIH staff are there as facilitators. As I said, I have industry experience, but the last thing I want to do is tell you what compounds to make, because I don't want to be an inventor on your program. I don't want to get intellectual property. That would defeat the program's purpose. So that's what we try to do. The consultants will also sign IP agreements with you. This is a great program. example of what NIH can do for a project from start to finish. Sue Slogenhoff was a basic researcher in familial dysautonomia. She identified the gene locus and the gene, carried it through, and worked with us. There was some special funding to screen a collection of compounds for NINDS and found kinetin. She developed a mouse model and eventually made her way, after working with Intramural, to the Blueprint as a program. And that project was brought in 2012. We took that project, did lead optimization on the work, eventually got to really good molecules. PTC Therapeutics came in, bought the whole project, lock, stock, and barrel from Sue, brought the whole thing in-house, and is carrying it forward. We have some videos up on our website, testimonials from both Sue and Paul Kenny about the program. And I think this next slide sort of will give you a flavor of what I'm talking about. We have had 22 projects to date. All the ones where you see the sort of green money signs, they've gotten outside investors to come into the program and provide funding to that program. And people, as I mentioned, PTC Therapeutics for the Slogenhoff project, Paul Kenny's project for smoking cessation, AstraZeneca has licensed. A project that actually stopped in BPN early on because it needed to develop animal models was able to take the compounds and still create a company and get funding for it. Our most recent announcement was from Blackstone Therapeutics that bought the migraine project from Scripps and has continued with the Blueprint. Tetra Discovery Partner was our first project for mild cognitive impairment. That one actually completed phase 1, is in phase 2 now. And NEI program was licensed by Lin Bioscience recently this year. Is going to be taking the project into the clinic as well. So the program certainly has had a bright past. It'll have a brighter future with you all, bringing me your great ideas going forward. I really, I'm out there to help you. If you need guidance, do contact me. I want to touch on a few other things now. The Translational Neural Device program is Nick Langhall's project. He works with Stephanie Fertig at Division of Translational Research. They just issued these new RFAs, and I would highly recommend you contact Nick about any translational device question. There's not only the translational neurodevices, but there's also other mechanisms out there. For instance, the Brain Initiative is putting out FOAs all the time right now with all the money that's in that program. I think they had 30-something FOAs last year, and they probably have a similar, if not more, number coming out this year. They take both invasive and non-invasive devices. I'll just touch on the small business programs, NINDS, just to mention that, it was, Matt covered it all. Every institute has it, all 9 mine too. But you may also be interested in a new program that NINDS, NCI, and NHLBI have just started up. We call it the Applicant Assistance Program. It's for those who have never received a small business award can apply for this program. It's free services intended to help you submit a competitive SBI or STTR. You must be within the mission of either NINDS, NCI, or NHLBI. And again, contact Stephanie if you want more information about that. The Brain Initiative, which I touched on, the most active funding is located at this website. As I said, there's so many FOAs coming out all the time that you really need to contact them regularly and keep up to date, because there'll be a large number of FOAs coming out during the course of the year. If you're in the device area, keep an eye out for that. Lastly, on clinical resources, we have a number of networks, the Stroke Network, StrokeNet, Neuronext, that can help you with late phase clinical trial work. We have Cywin and NET, which can help with emergency treatment trials, both of which are active and ongoing. Clinton Wright is the director of the Division of Clinical Research and is a good contact for that. He can direct you in the right direction. I promised to give you some highlights of other services available around NIH, so I will. Preclinical development, the Alzheimer's disease drug development program is a U01 program, and it's designed to give up to 5 years of funding for both devices and small molecules and biologics. I would contact Larry Ruffolo. Larry will direct you. Larry's also the program officer for Blueprint, whether you think your program is most appropriate for Blueprint or for the NIA specific funding. The Cooperative Drug Discovery and Development Group does treatments for mental disorders, drug and alcohol addiction, and can provide additional, again, significant funding associated through either U01 or U19 mechanism. And there are a number of contacts listed here depending on which institute you're interested in. We have strategic alliances for medications development to treat substance abuse disorders. NIDA puts this out. Yvonne Montoya is the acting director of the division there and can certainly help with this R01 as well as other opportunities that they have at NIDA for this. And they just issued an FOA in December, I think right around Christmas time, for one around pain. And another U01 mechanism that, again, takes all modalities and takes it up through clinical trials and will complement the BPN if you're in the pain area. I mentioned the epileptic therapy screening program. There's also the addiction treatment discovery program, which screens compounds and animal models of addiction. It's important for people in the pain area right now. It's important for people working in the drug addiction area. The psychoactive drug screening program, Brian Roth's program out of UNC. If you don't know what it is, it's a great resource. Again, it provides screening of your compounds against a number of receptor assays and can give you a lot of information there. And I believe it's free for the asking. It is well subscribed, however. Services in toxicology. NIMH has a toxicological evaluation of novel ligands program. Jamie Driscoll is the contact person for that. NIDA has one as well, and Nathan is the contact for that one. And Bridges out of NCATS is a program, I've heard it referred to at times, sort of like summer camp for your molecule or your program. Is they'll take it in and they'll do all the work in terms of getting it ready for an IND. There's a caveat, which is they're also going to become inventors, and NIH will have intellectual property in some cases, depending on what you're doing with them. And they also do chemistry support. So if you're not only toxicology, if you're doing toxicology, they probably won't have any IP, but if you're doing chemistry, they will potentially have that. NIMH has a synthesis and drug supply program. NIDA has a program in their chemistry and pharmaceutical experience, also for doing things like medchem, analytical work, and Nora can help you with that. I think those are all the services I'm aware of that can add on, and you can touch these, add these pieces and these resources to your program to get the data you need to get you ready for another FOA, get you the data you need to get you ready for the SBIR program to be competitive for that, hopefully direct to phase 2 when it comes back and things like that. So there's a number of opportunities. Being as we have a little time, I'll go through my general consideration slides. The first thing I want to tell everyone to do is build a collaborative team. It's probably the most important thing you can do. Involve your clinicians early. Get the people to really understand the patient population to inform you on what your target product profile should look like and what experts you need to have in disease biology. Make sure you really talk to your business development people early. It's important to understand and get them on board because NIH is not going to pay for you to file your patents, and it's not going to pay for the national filings to keep them going. And so you need to make sure you have that support to do that. If you're working with an academic, your tech transfer office, or your business development people, make sure they get the money in place to do that, because that's going to be essential for when you go to license it down the road. Consider partnerships with biotech if you're an academic type person as well. Plan with the end in mind. Know what your target target population is. Is it pediatric? Is it adult? Is it early? Is it advanced disease? Know what your dosing regimen's going to look like. Is it acute or chronic? Know what route of administration it is and what your desired outcome. So it's really important to engage your clinicians as early as you possibly can to do it, because you have to develop your target product profile, and that's going to help you figure out what institute to go to. It's going to be essential to figure out which program director you need to talk to get them to buy in. Because for me to get a translational program, as is the truth for all the translational program directors, by our council, by our IC management, I have to have a program director on the disease biology area standing right next to me saying, they agreed, this is a great project too. And part of that is knowing that you're in the right area, that you've got a good competitive project, that you're going to impact patients. It also helps you define clearly what institute you're going to be in. Is one thing, you can have dual assignments, but on these large projects for drug development, it's oftentimes difficult to get multiple people to assign to it. Usually they want to say, you're going to be impacting my mission, and therefore I'm willing to pay for it. Also recognize hit compounds are not clinical candidates. And when you get reviewed, if you don't make the effort to explain that in your application, if you leave it to the reviewers to figure it out, they will not. They will assume you don't understand the difference and will negatively score you because of that. So make sure you're addressing things like it's a hERG inhibitor, or it has off-target effects, or is an inhibitor, or I don't own the IP of it, or is it consistent with what my plans are? All of those things are really important. In a 12-page application, you got to be concise, but you've got to cover everything, and so that they really understand that you've thought about this project well. It's a drug development project. It's going to be lots of money. Make sure that it comes across that you're thinking about all the issues and done some of that competitive assessment, dotted some I's, crossed some T's, made sure you understand it. Incorporate IP into your strategy. Consider the future licensing strategy. Understand that you don't develop someone else's compound without permission, and avoid encumbering your own work for future work. So if you're going to publish all of your SAR before you come in, and that's going to be a priority, a prior art, you're going to have to explain how you're going to get around that prior art to develop your actual drug compound. So think about that before you go off and publish all of the work that might lead up to it. You can label in the applications the research strategy as business confidential, the part of information for which is clearly not been published anywhere and you don't ever expect it to go out anywhere. But do include the data that you think is important. So don't not include structure, for instance, because you're worried about NIH is going to disseminate structure. We're not. You just put it in there. That'll be an extra added layer of protection against a FOIA request anywhere down the road. But the reality is we don't publish applications. Consider your scientific premise. Explicitly discuss the data being presented both from your work that you've done and the prior publications in a detailed manner. You need to really discuss the rigor of it. Is it randomized? Was the data blinded? Was it, sample size powered correctly? Was there opportunities for bias that weren't controlled for? Do it for both your work and anything you're citing as the preliminary data that's going to be part of the scientific rationale for why this program is important. Complete your registrations. Matt pointed this. I can't tell you how many times I've suffered through this with people trying to get them through at the last second. And it's worked in some cases. We've gotten them through, but it's not in our control. We can help you fill out the application if we have time and hold your hand through some of the process, but ultimately it's up to you guys. Consider applying early. And I'll give you, for instance, from last year, a notice came out in January that said if you put something in the appendix, it's not called for in the FOA, the application will be withdrawn. A week and a half later, my FOA was due, and I had 3 or 4 applications that were drawn by the review, or by the receipt and referral, because there was something in the appendix. There was nothing I could do. So if you submitted it early and you got that error, you could adjust it. But if you submit it at the last second and you get an error like that, they're going to just, there's nothing you can do. Check your budget limits and permission requirements. If you're going to go over the 500K, you've got to give me plenty of notice to go through that process. And as I said, talk to your tech transfer and business development people. You need to have a plan, and especially for the BPN, it's a requirement of the FOAs to have the discussion of your IP strategies. I guess one of my last couple of slides here deals with training in neurotherapeutics and discovery and development. It's just a plug for a course that's being at the Ascent meeting that is part of one of my the Blueprint program that we have. This training course this year is both biologics and small molecules. And the BPN, because I'm the BPN program director, the receipt date is February 7th, just a few weeks down the road. It's reviewed by a special emphasis panel, not by CSR, and both groups are reviewed simultaneously. They review all the discovery projects first, then all the development projects next. So that's it. I have time for questions, and thank you for your attention. Oh, sorry.
You had a couple of services slides on chemistry.
Yep.
Are those standalone projects or are they in the context of?
I believe those are all standalones. The ones I had for MedChem chemistry, Bridges is an intramural program, and that's the one I said you sort of hand your project over to them at NCATS, and they'll work on it. They have their own MedChem intramural scientists doing research on site there. The NIMH drug supply program, that's not, I think it's available to small businesses and anyone in the community can contact them. NIDA is the same way. Again, they have contract support. This is done through contracts. Each FOA is different. So I can tell you for Blueprint, through the UG3, UH3, you can apply as a foreign institute. Through the Blueprint for small molecules, if that's what you're interested in, then the UG3, UH3 will accept foreign. Create, again, I think the non-SBIR track does.
For these services, what does the process look like for applying them? What is the timeline?
For these kind of services, you would need to talk to them. Some of them are you just submit your sample and you get into the queue, and sometimes the queue is longer than others. Others, like Bridges, I think is an application process they do a few times a year where they review applications, they accept the programs they like, and they move forward on it, so it's competitive.
Do you see AR trials using AR for dementia or this kind of technology drives changes in security clinical?
I'm not 100% sure what you're asking me. Sorry, can you try it one more time? I missed the beginning of it.
Some consumer space.
Consumer.
Using AR.
Oh, augmented reality, yes. I think there are some opportunities there. I would contact Nick Langhals at NINDS, who's a device person. He could probably direct you, either him or through the BRAIN initiative, to be more plugged into that opportunities there than I would be. I'm sorry.
For a biomarker study, is it best to go after an omnibus under NINDS or to go after Alzheimer's if it's across neurodevelopment or NIH?
I would say with biomarkers, you probably be best going through the omnibus presently. I will say, what I can say is biomarkers are a hot topic, and we are developing our strategy along those lines. There are some FOAs out there that will help with biomarkers. You just have to search for them, but I suspect there'll be some specific ones coming.
Will the program IC1 is it applicable for phase 2 clinical trial Alzheimer's? Basically, we like to do the phase 2A, and we have to do dynamics and technology and so on. phase 2A
Yep.
program to support that, or it is all new?
I'm sorry, which program are you asking?
The one you mentioned, line 37.
Okay, so the blueprint, no. So blueprint, no. Blueprint will only do through phase 1. Things directed at phase 2 and beyond, you would need to contact either NIA or the NeuroNext program. Because Alzheimer's disease is a shared portfolio between NIA, aging, and NINDS and neurology, it would depend on who wants to fund it, but there are funding opportunities to do phase 2 trials for both, at both ICs.
So we can do to expand the pharmacodynamics or pharmacodynamics.
Not anything directed towards beyond phase 1 is out of scope of Blueprint. Blueprint's really designed to get to that first in human experience. It's actually a gap I pointed out to a number of people at NIH, hoping that we'll eventually develop some mechanisms around it, but it's very expensive to do things like advanced PK for supporting phase 2, running the long-term toxicology trials, or GLP tox studies, I mean. So I think the best place to try to do those is probably through the omnibus SBIR and make an argument as fee for service. Sure, let me go back and I will. Which page? Sorry, one more.
The page before the last page.
Before this page, this toxicology?
So the last page, go back to the last page, and then go back one page.
The very last page
Yeah.
that I talked about, sorry. And? Oh. I'm trying. This one, there we go. Okay.
You talked about a goal you have every 2 weeks on a blueprint
Yes.
program. That sort of begs the question about, as a sort of drug developer, I know I like to kind of get into things even when I probably shouldn't.
Mm-hmm.
What is your sort of client satisfaction rate? Do you have some sense of whether you guys tend to get over-involved, or how do you?
Well, I mean, we meet every 2 weeks as a team. And as I said, for the small molecules, this is true for the Blueprint Neurotherapeutics. It's not true for Create. They tend to meet fairly infrequently because all the work is being done there. Our 2 weeks is really to coordinate contracts and all the other activities going on. But the lead for that is the PI and the lead consultant. I expect them to create the agendas and to drive the discussions. If they're working through our contractors, it's an opportunity for the contractors to report back on what they've done. Here's the medicinal chemistry, here are the analogs we've made, here's what are we going to test, which compounds are going to advance into the next set of testing, which ones are going to go there. That's what we're doing in most of those meetings. It's also getting ready for when you're meeting coming up on a milestone decision. Have we done all the right experiments? You have the right expertise in the room. When we're designing a toxicology study, is it the right toxicology study? Is it the right doses that we're picking? Have we figured this? Have we dotted our I's, crossed our T's, so that when we go to do a milestone review, we're going to do well. And I should mention the Blueprint Neurotherapeutics Network has an external oversight committee that's people who are outside of NIH, industry experts that have both experience in the pharma sector, in academic drug discovery centers, and venture capital. And so they can give you sort of a flavor. Do they think you really met your milestones? Is your target product profile appropriate or not? Would they consider other things that they'd want to hear as investors in the program? You also got to think of, as we're moving through these projects, NIH is an investor with you, right? Because we're spending millions of dollars in tranches on you as you go through. And so we want to know that everything looks good too. Okay, thanks.